This is the first set of notes on genetics. In today's lesson, we'll talk about Mendels Laws and how the science of genetics got started. Vocabulary terms, there are a lot of terms in this. A lot of them are new terms, and so you'll need to pay close attention as we go to make sure that you pick up on the terms as we go through. So first of all, we'll talk about Mendels Laws. You know, Mendel was a monk in a monastery, and he figured out a lot of stuff about heredity. He was studying heredity of garden peas. Heredity, by definition, is the transmission of traits from one generation to next, and genetics is the study, or the scientific study of heredity. And so he started in the 1860s breeding garden peas and was looking at the different kinds of peas that were available. As a result of the long-term studies that he did of pea characteristics, he was able to come up with some conclusions about how things are inherited. Before Mendel's research, there was a lot of misunderstanding or lack of understanding about how heredity occurred. A lot of people thought that the inheritance of characteristics was going to be a blending of characteristics of the two parents. What Mendel decided, because of the research that he did, was that there are individual inheritable factors or heritable factors which are going to be inherited from the parents. Some of those factors can be exhibited while others might be hidden, but show up in a later generation. So he called a heritable feature that varies among individuals in a species a character. So in the first example that we'll give of Mendel's research, we'll talk about flower color. Each variant or difference of the character, such as the purple flowers or white flowers, is called a trait. And so we have adopted some of his language, and then other things that he described, we have changed the wording because of our later understanding of what was happening in the cells. This is Gregor Mendel, who was, as I said, a monk in a monastery. He eventually became the abbot of the monastery. He was a teacher. He studied mathematics. He was a very strong background in mathematics, and also studied science in university, and wanted very much to teach. What he found was that there were different varieties of peas that were called true breeding. This is what results when the pea flowers self-fertilize and they produce offspring that are all identical to the parent. If you have two different varieties and manage to cross-fertilize or cross-pollinate them, then you would produce something called a hybrid. And so this is the description of what we call a genetic cross. The terminology that Mendel used, that we will also use and still use today, is calling the true breeding parent generation the P generation. The hybrid offspring of two parents with different traits would be the F1 or first filial generation, and then a cross of two F1 plants would produce the F2 generation, just so you know what the terminology means as we go through this. Here's what he did. Okay, the pea flower here has the stamens and the pistil. It's a complete flower, so it has all the parts right there together. There's usually another petal that kind of covers this over, which is why they generally self-fertilize. And so what he did is he cut away the stamens from the purple flowered plant and took a paintbrush and transferred pollen from a white flowered plant to the carpel of the purple flowered plant. The carpel was then fertilized and pollinated and eventually developed into the pea pod, or matured into the pea pod, and the pea pod contains the seeds that result from that cross. When those seeds were planted, what he found was, instead of having pea plants with light purple flowers, they were the same purple as the original purple flower plant, and all of them were purple and none of them are white. So it looked as if the white characteristic had disappeared. There were seven different characteristics that Mendel looked at in peas. He looked, of course, at flower color, the position of the flower, whether the flower was on the sides of the plant or at the tip of the plant. It's called axial versus terminal. The seed color, either yellow or green. The seed shape, either round or wrinkled. the pod shape, inflated or constricted, the pod color, green or yellow, and the stem length, either tall or dwarf. We'll call that tall and short. Notice in the chart that you just saw, the dominant characteristic is on the left and the recessive is on the right. So what he found was when he crossed two individuals differing in that single character, flower color, that's called a monohybrid cross. Mono means one, and hybrid is the cross between parents with different characteristics. Mendel did this cross on the plant with purple flowers and the plant with white flowers, and like we saw in the pictures, the F1 generation produced all plants with purple flowers, but when he crossed F1 plants with each other or allowed them to self-fertilize, what he found in the F2 generation was that 3/4 of the plants had purple flowers and 1/4 had white flowers. So here's the results that he got. His P generation were true breeding plants, and then when he cross-pollinated them, all the plants that resulted from that crossing had purple flowers, and then when those fertilized themselves or fertilized each other, 3/4 of the plants had purple flowers and 1/4 had white flowers. So the white characteristic did not go away, it was just hidden for a generation. He had to figure out a way to explain why the white color seemed to disappear and reappeared later on in a different generation. And so he observed this pattern of inheritance not only for flower color, but also for the other 6 characteristics that he studied in the time that he was studying peas. As a result of the work that he did, he planted thousands of plants, counted thousands of flowers and plants and peas. Okay, he developed some hypotheses using about what's happening, and these are restated here in modern terminology rather than the terminology he used. First of all, the alternative versions of the genes are called alleles. So that would be a purple allele that would produce a purple flower, and a white allele that would produce a white flower. For each characteristic, each organism inherits 2 alleles, one from each parent. The alleles can be the same or they can be different. If the 2 alleles are the same, they would be called homozygous. Homo means the same, and zygous refers to the zygote, the fertilized egg. If the 2 alleles are different, then that would be a heterozygous genotype because it has 2 different alleles. Hetero means different. If the alleles are different, then one of them is going to determine the appearance of the plant. That one's called the dominant allele. The other doesn't have an effect on the organism's appearance. That's called recessive. And so we're talking about the phenotype, which is the appearance or the expression of the trait. The genotype is the genetic makeup, the purple allele, white allele. And the one thing that he did find sometimes, or that we have discovered since then, that the same phenotype can sometimes be determined by more than one genotype. We're going to talk about the more simple inheritance where they're more related just one to one or the other. Another hypothesis of Mendel's that resulted from Mendel's work is that a sperm or an egg carries only one allele for each inherited character, because here's what happens. As we know, during meiosis, those allele pairs are going to separate from each other. He called it segregate, which just means separation. They're going to separate during the production of gametes. This statement is called the law of segregation. Mendel did not know about meiosis, but now that we know about meiosis, we can use that to help explain why each sperm or egg cell only carries one of each pair, one allele of each pair. This also explains that 3-to-1 ratio in the F2 generation. Okay, remember that all the F1 hybrids had a heterozygous genotype, the capital P standing for the purple flower, the purple color trait, and the lowercase p standing for the white color trait. We can use a Punnett square to show the different combinations that could occur when you combine those gametes. Okay, so now let's look at the diagram again, looking at the alleles and a Punnett square. Okay, so here are our parent generation with our true breeding purple flowered and white flowered plants. Every gamete that is produced in the purple flower would have that capital P, or the dominant purple allele. Every gamete of the white flowered plant would all have the lowercase p, or the white flowered allele. When those two gametes combined to make the F1 generation, then all of the plants in that generation would have a big P for purple and a lowercase p for the white. But they look purple because the purple masks or covers up the white. The purple is expressed, the white is not. When these plants produce gametes, their gametes can have two possibilities. The half of the gametes would have the capital P, the purple flowered allele, and the other half would have the lowercase p or the white flowered allele. So now we have to show for the F2 generation what happens in the formation of the zygotes. So the letters on the outside of the Punnett square, the circles here, represent the gametes that are produced by the plants. So from the F1 plant, we would have from one flower, we would have the capital P and the lowercase p here, and from the other one, the eggs, we would have the same characteristic. In the Punnett square, we're going to combine the two, the row and the column to give us our genotype of our offspring. And so here we have two capital P's. Here we have a capital P and a lowercase p, the same thing here, and the two lowercase p's. And that gives us our 3-to-1 ratio that he saw in the F2 generation, because 3 of these have the purple flower allele, which gives them the purple color, and 1 has only the white flower alleles, which gives them the white color. The ratio in the phenotype, or the appearance, is 3-to-1, 3 purple to 1 white. Genotypes, we have 3 different genotypes. We have homozygous dominant, 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive in the offspring. One thing I always like to remind students about is that the Punnett square isn't a prediction or a promise of what's going to happen. It is a probability of what can happen, and each Punnett square represents the things that can happen for each separate fertilization event. So every fertilization event is totally separate from every other one, and it doesn't predict what will happen in terms of all of the offspring, but only of that one fertilization event. Now, one thing that we need to talk about when we talk about the alleles is homologous chromosomes. Of course, the homologous chromosomes can carry those alleles for the different characters. A locus, or the pluralis loci, is the specific location of a gene on a chromosome, and the word homologous means the alleles reside in the same location. So, and if you're homozygous, you have the same allele on both homologous chromosomes, and if you're heterozygous, you have 2 different alleles on the homologous chromosomes. Here we have 2 homologous chromosomes. Okay, there are 3 alleles that we see on each one. Okay, the gene locus is here, here, and here. And so when you look at both chromosomes, you can tell that this individual is homozygous dominant for the P allele, for the P gene, homozygous recessive for the A gene, and heterozygous for the B gene, whatever it happens to be. You can also look at 2 characters at once and perform something called a dihybrid cross. Di means 2, okay? And so we're going to look at mating parental varieties that have 2 different characteristics rather than just 1. And so this is one of the dihybrid crosses that Mendel performed. He crossed true breeding plants that had round yellow seeds. The round shape is one gene, and the yellow color is another. And he crossed the round yellow seeded plant with a wrinkled green seeded plant. In the F1 generation, all the plants had round yellow seeds, which indicates to us that round is dominant over wrinkled and yellow is dominant over green. The F2 generation, when they were allowed to self-pollinate, he found some very different results. He found that 9/16 of them had round yellow seeds, 3/16 had wrinkled yellow seeds, 3/16 had round green seeds, and 1/16 had wrinkled green seeds. So let's see how that works. Here's our parent generation. Now, remember that you have 2 alleles for each characteristic or character, and so we've got 2 R's for round and 2 capital Y's for yellow for the round yellow seed, and then we've got 2 lowercase r's and 2 lowercase y's for the wrinkled green seed. Each gamete that is produced is going to contain an R and a Y. one of each. And so for this particular plant, the only type of gametes that can produce have one capital R and one capital Y, and for this one we have a lowercase r and a lowercase y. When those combine together to make a zygote that produces the F1 generation, we're going to have heterozygous for both traits. They'll still show the dominant traits, but they're heterozygous. They do carry the alleles for the recessive. Now, if those genes were inherited together, we would expect the cross to look something like this. We would see, if we could see that everyone that had this would have, if they were always together, then we would have 3/4 with yellow round seeds and only 1/4 with wrinkled green seeds. But that's not what he found when he did the cross. It was very different. This is what he did, okay? There were 4 possible combinations of the alleles, and we do the same thing that you do in math, that FOIL technique, the first, outside, inside, last, when you're multiplying binomials. So we have capital R and capital Y is one dominant for both alleles, is one possibility. Recessive for the first and dominant for the second is another possibility. Dominant for the first and recessive for the second is a third possibility, and recessive for both alleles is the fourth possibility. When you show these different possibilities, then you can see that if you do the crosses, you'll see that 9 out of the 16 possibilities are dominant for both traits. 3 of the 16 are dominant for the color and recessive for wrinkled. 3 are dominant for round and recessive for green, and 1/16 is recessive for both traits. And this is what supported the hypothesis that Mendel came up with. He needed to explain why this happened, okay? And so he suggested, this is another one of his principles or laws that we follow, is that the inheritance of one characteristic has no effect on the inheritance of another. He called this the law of independent assortment because these 2 alleles were independent of each other. They did not depend on each other to get their trait. The figures below are just going to show us what happens when we look at some other kinds of organisms. Instead of pea plants, we're now going to look at Labrador retrievers. So there are 2 traits we're going to look at in Labrador retrievers: black coat color versus chocolate, and normal vision versus progressive retinal atrophy, which eventually produces blindness in the dogs. This is a recessive condition that occurs in Labrador retriever dogs. So here we have possible genotypes of the dogs. We can have one with black coat and normal vision, one with black coat with the PRA or the blindness. We could have chocolate coat and normal vision, or we can have a chocolate coat with the blindness. So when you cross 2 dogs that are heterozygous for both traits, here's what you find. When you cross those over all the puppies that are born from that union, we would have 9 of the 16 would have a black coat with normal vision, 3 of the 16 would have a black coat with blindness, 3 of the 16 would have a chocolate coat and normal vision, and 1 of the 16 would have a chocolate coat with blindness, just like we saw with the peas. Okay, the way you determine an unknown genotype is by doing something called a test cross. This mates an individual that has the dominant phenotype but an unknown genotype with a homozygous recessive individual, and this can show us whether the unknown genotype includes a recessive allele. And this is what Mendel did to figure out that he had to verify that he had true breeding genotypes. So let's look at how we would use that to determine the genotype of a lab with normal eyes. Okay, so we have a black dog here with normal eyes. We're not going to worry about the eye condition here. So we have a black dog, and we're going to mate that with a brown dog or a chocolate dog. There are 2 possibilities for the dogs. We can produce puppies that are all black, which would indicate that the black dog was homozygous for the black coat color, or if we produce some puppies that have chocolate coat, then we would know that the black dog was heterozygous for that characteristic. One thing that Mendel had was a very strong background in mathematics, and he used the laws of probability to determine what the outcomes were. So you can look at the way probability works and kind of figure out what's going to happen out of the total possible outcomes. When you figure out the probability of two independent events, this uses the rule of multiplication, in which the product of the probabilities is going to give us the final probability. And the probability that an event can occur in two or more different ways is the sum of the separate probabilities. This is called the rule of addition. Here we have two coins being flipped. One is a dime, one is a penny, and each one has a 1/2 or 50% chance of coming up heads or tails. If you want both coins, if you want to know what the probability of both coins coming up as heads, that would be 1/2 times 1/2 or 1/4. The same thing with heads for the first and tails for the second, and so forth. And using probability helps us determine what the probability is of getting a particular outcome.